Okay, so genes might not necessarily follow these Mendelian characteristics. If you have a gene that has more than 2 alleles, like for instance eye color, you can have green eyes, brown eyes, hazel eyes, combinations of both. So those necessarily aren't going to follow that simple genetic or Mendelian pattern of inheritance. And also another reason why some genes might not follow Mendelian inheritance is because that one gene is capable of producing multiple different phenotypes. So instead of tracking one trait for purple and you only seeing one result, you might have one trait that can give you multiple different results. So that complicates everything. So in other words, some different traits might not show these Mendelian types of inheritance. And so we're going to talk about some of the alternatives to Mendelian inheritance. And a lot of it has to do with how dominant the trait is going to be. So we mentioned that in the purple flower experiment, as long as a flower received one of those dominant traits, a capital P, as long as it received that, it was going to be a purple flower. So we call that complete dominance. So in other words, you only need one copy of that dominant trait to see the dominant phenotype. And that's what explains why we see purple flowers when you have a big P, little p, or a heterozygote. Is because in complete dominance, you only need that one capital P in order to show you the dominant phenotype. Now, some traits, they do not show complete dominance. Some of them can show incomplete dominance, and then some of them can show codominance. Incomplete dominance is where you still have a you still have a dominant trait and a recessive trait, but that dominant trait isn't going to be as strong as it was in the purple. So an example of this is if you take a flower that is red and you cross it to a flower that's white, you end up getting a pink flower. So even though red is supposedly dominant, it's not completely dominant over the white allele. And so in other words, you kind of see this mixing or a little bit of both of the parents. So red flower times white flower gives you pink flower and not red flower, white flower. And then the other type of deviation in dominance is called codominance. And codominance is where you have 2 alleles that both contribute to a phenotype independently. So we've been talking about the purple flower allele codes for the purple flower, and the white flower allele codes for the white flower. Well, in the case of codominance, we have 2 different alleles, both contributing to the phenotype. And so an example of this is actually going to be blood type. Your blood type is based on these little proteins or sugars that are attached to your red blood cells. And the AB blood type, that's actually 2 different alleles. There's the A allele and the B allele, but they're both contributing to the AB phenotype of your red blood cells. So if you look at the example of incomplete dominance, again, we're taking a red flower and we're taking a white flower. And these are, in particular, snapdragon. So if you know what a snapdragon flower is, they're always going to exhibit this incomplete dominance. And so if you look at the genotype, again, we took true breeding red flowers and crossed them to true breeding white flowers. So in other words, homozygous dominant red to homozygous recessive white. And when we cross them, we get one allele from the red plant and we get one allele from the white plant. And so we end up making these heterozygous plants that are going to be pink. They're going to show this kind of intermediate phenotype between the original parents. And then when he did a monohybrid cross and he crossed the F1s together, in other words, he crossed a pink to a pink, the possible combinations of alleles, he's either going to have an R allele or a W allele at that particular gene. And so when you look at the Punnett square, you're in the F2 generation, you're actually going to see a different phenotypic ratio. So you're going to see the homozygous dominant come back and show you the red flower. you're going to see the heterozygous, which is going to show this incomplete dominance. Another heterozygous right here. And then when you have the two recessive alleles coming together, W and W, that's going to give you back your white flower. So you don't totally lose the dominant traits. It's just when you cross a true breeding red to a true breeding right, that first generation, all you're going to see is pink. But then when you cross the F1s from that, and you cross a heterozygous to a heterozygous, that's when you're still going to see that 1 to 2 to 1 ratio. So 1 homozygous dominant, 50%, or sorry, 2 heterozygous, and then 1 the homozygous recessive. Questions? All right, so when we're looking at codominance, again, codominance is when you have 2 different alleles or 2 different traits that contribute to a phenotype together. And so blood typing is the standard example that we use for this. And so when you're talking about blood type, your blood type is determined by a sugar molecule that's been attached to your red blood cells. So your sugar molecules can either be the A-type sugar molecule, the B-type sugar molecule, or there's no molecule of sugar at all. And so depending on which of those sugar molecules are present on your red blood cells, that dictates what type of blood you have. And so blood groups that are going to only have this A-type carbohydrate are going to be type A blood. Those red blood cells that only show this B-type carbohydrate are going to be type B blood. And type AB blood is when you have both of these sugar molecules present. So this is the example of codominance right here. You have 2 different alleles that are both present, that are both contributing to the phenotype of the cell. And then people with type O blood, they're not going to have any of these sugar molecules present. And so you'll know that type O is the universal donor, right? The reason why is because blood typing has to do with your immune system. So your immune system, the way it functions, is it goes around looking for stuff that it doesn't recognize. So if you have a person that has type B blood and you give them type A blood, then your immune system's not going to recognize this type A sugar. So when it's used to seeing all these type B sugars. So as a result, your body will start rejecting that different type of red blood cell because it recognizes these A-type sugars as being foreign. But if you take a type O, you can give it to any of these other blood types because it doesn't have any sugar molecules to recognize. So therefore your immune system isn't going to be threatened by this because it just looks like a normal red blood cell. So that's why type O blood is going to be the universal donor because it doesn't have any of these carbohydrates or sugar molecules attached to it, so it means it can't be rejected by your immune system. So you can give it to type A, type B, or type AB.
If you have AB, can you get just A or just B?
Yes, you can.
Because it'll match parts of it.
Exactly. AB is going to be the universal acceptor because they can receive type A and be fine. They can receive type B and be fine, and then they can also receive type O and be fine. But you could not take a type A blood cell and give AB because that B is going to be foreign, and again, your immune system is going to recognize that and start to attack it. And so when we write out these blood phenotypes, we always use a capital letter I don't ask me why, and then it shows the type of carbohydrate present. So if you can have a homozygous dominant red blood cell, so you would have capital A, or you can have a heterozygous, in which case you write the capital A with the little I. And the little I says that there's no carbohydrate present. And so the same thing with B. You can have big B, or big B with a little I. And then the AB, they're automatically going to be big A, big B. And then the type O is going to be little I, signifying basically the homozygous recessive version of the phenotype.
So O can be given to any of them, but
And-
the other three can't be given to O, right? Or is that
Right, you can't give anything to O.
to O?
Because, if your immune system isn't used to seeing any carbohydrates, so if you now introduce an A or a B, your immune system's going to freak out and attack the cell.
Okay.
So O is going to be the universal donor, AB is going to be the universal acceptor.
Okay.
So you want to be AB. So if you get in a car crash, you go to the hospital, it doesn't matter what kind of blood they have, you can take it all. All right? And you don't want to be type O because then everyone's always hitting you up for blood, right? All right, any more questions about this? Okay. Some other non-Mendelian inheritance patterns that can be observed is called pleiotropy. And so pleiotropy is when you have one gene that's responsible for multiple different phenotypes. And so a lot of diseases actually show this pleiotropy. Cystic fibrosis, for instance, is a genetic disease that is caused by a mutation in only one gene. I forget the name of the gene, but it's called the CF gene for cystic fibrosis. So if you get a mutation in this gene, you're going to end up with cystic fibrosis. But there's not just one phenotype associated with cystic fibrosis. You're going to have muscle issues, you're going to have cognitive issues, you're going to have breathing issues. So these multiple different things that are going to go wrong just from a mutation in this one gene. That's what pleiotropy is. Another example is sickle cell disease. Sickle cell is caused by one mutation in one gene, but you get all of these other phenotypes that are associated with that disease. And so this is what we call pleiotropy. And so when you have these genes that are pleiotropic, they're usually not going to follow these Mendelian inheritance patterns. Another kind of non-Mendelian type of genetics is called epistasis. And what epistasis is when you have two different genes that are both controlling a phenotype. But one of those genes is going to be more dominant than the other. But nonetheless, you still have two different traits contributing to a phenotype. And so one example of this is Labrador retrievers. Y'all have seen black labs and chocolate labs and white labs. Well, the color of a lab is determined by two different genes. One gene determines what color the animal's going to be. So brown being dominant to black. Or I'm sorry, black being dominant to brown. So that's why we see more black labs than we do brown labs. But there's another gene that determines whether or not that pigment is going to be deposited in the animal at all. So you have this secondary gene. It doesn't matter what the coat color is, brown or black. It doesn't matter if that second gene says that pigment isn't going to be deposited at all. And so we say that the gene for coat color is going to be epistatic to the one that dictates pigment. And so if you look at the different possibilities, so in this case we're taking heterozygous black labs. They're heterozygous for the coat color. So in other words, they're going to be black because black is dominant to brown. And then for the other gene, for this E gene, that determines the pigment, whether or not it's going to be deposited. So in this case of the parentals, deposited color is going to be dominant over non-deposited color. And so in this example, you could see, or in all of these, I should say, you can see that the dominant trait is going to prevail. So in other words, because you have one of these dominant alleles of coat color, one capital E, that's going to determine that you are going to deposit whatever this genotype says. So in other words, if you get an organism or a dog that has little e, meaning it's homozygous recessive for whether or not you're depositing that color, it doesn't matter what the genotype of the coat color is, because that means that color is not going to be deposited. And so if that color is not going to be deposited, that's how we get yellow labs. So a yellow lab can have a genotype for black, big B, or big B, little b. But if that other gene is homozygous recessive, it's going to automatically be a yellow lab. So that's why we say that the coat color, the gene for coat color is dependent on the gene for if that color is deposited. So we say that coat color is epistatic to or depends on the expression of this gene. And so the way that we get chocolate labs is if you have these, the coat color gene is present, so in other words you have a capital E that's telling you that that color will be deposited, and then if you have a homozygous recessive gene for coat color. In other words, little b is going to determine that you have a brown lab. And because you have at least one copy of that second gene that determines if it's going to be deposited or not, that's why you're going to see a brown lab. Questions about epistasis? All right, we're almost done. Okay, so another type of non-Mendelian genetics is called polygenic inheritance. And polygenic inheritance is when you have multiple different genes that are going to contribute to a phenotype. And so these are the kind of tricky phenotypes like skin color. It's not like there's a black, a brown, and a white skin color gene. You know, it's a whole different range of phenotypes going all the way from light to dark. And so there's multiple different genes that contribute to skin color. And so these genes, think of them as kind of like levels of pigment. And so there's kind of an additive effect. So in other words, if your 6 genes all have the dark pigment, your skin's going to be really, really, really dark. If all 6 of your genes show a light version of pigment, then you're going to be really, really light. But most of us are kind of somewhere in between. And so it's depending on how many of these pigment genes you have. So people that are really, really, really light-skinned, they're going to have none of these, dark pigment genes. But then people who have all 6 that are dark colored are going to be extremely dark. But those are going to be the rare phenotypes. Most of us, like we said, are going to fall somewhere in the middle. So depending on how many of these dark pigments you have, then that's going to determine your skin tone. Another example of polygenic inheritance is height. All right? It's not like if you take someone who's 6 foot and cross into someone who's 5 foot, you're not going to get someone who's 5'6" in between. It's going to be, again, these units of tallness that are controlled by multiple different genes. So there's not like there's a gene for tall and a gene for short and a gene for medium height. It's a whole different phenotypic range. And so depending on how many of those, pigment units or how many of those tall units you get is going to dictate how tall you are or the color of your skin. So polygenic inheritance is when you have multiple different traits or multiple genes affecting one phenotype. So in this case, the phenotype is skin color, and the genes that we're looking at have various different amounts of pigmentation associated with them. So the more pigment genes you have, the darker you're going to be. The less of these pigment you have in your genes, the lighter you're going to be. And so most people are going to kind of fall in this middle range, or about half dark and half light. Questions? Okay. All right. So, one thing that we haven't talked about is the relationship between the genetics and the environment. So in other words, the nature-nurture argument. So nature says that your genes are going to dictate your behavior and dictate, your life essentially. And the nurture part says that your environmental factors can also influence the way you behave and how you act. And so this is going to be true in certain types of organisms. So hydrangeas are a type of flower. You can see them in the grocery store a lot for sale. And they come in multiple different colors. But their genotype is going to be exactly the same. So in other words, it's not like this is homozygous recessive and this is homozygous dominant. The genotype is the exact same. But you can get different colors depending on the amount of acid in the soil. So even with the exact same genotype, you can get 2 different colors. So in other words, it's that pH in the soil or that pH from the environment which is dictating what the phenotype is going to look like. And so we call this kind of the norm of reaction. So the norm of reaction is all the different phenotypes you can see based on a certain environmental condition. In this example, we're looking at hydrangeas and the acidity of the soil that they were grown in. They were grown in a higher pH, you might get pink. If they're grown in a lower pH, you might get purple. And so this is an example of the environment having an effect on the phenotype of an organism. All right, last little bit and then we'll wrap up. So we talked about these different traits and how they're inherited. Well, we can actually trace these different genes throughout a family using a pedigree. And what a pedigree is, it's basically a family tree that are tracking a particular trait or tracking a particular gene and determining and using that information to determine whether or not offspring are capable of receiving those traits. And so this pedigree analysis is going to be really important as far as following genetic diseases. So if you have a family tree and you say, oh, grandma had this disease and dad had this disease and my brother has this disease, what are the chances of when my brother gets married and has a kid, he's going to pass it on to his child? And so this pedigree analysis is comparing different versions of alleles for different traits. And so here we're looking at widow's peak. Widow's peak, you get that little in your hair. Well, you can look, and so all of these pedigrees are going to be set up the same. Squares are always going to be males, circles are always going to be females. So for instance, we took this male, married this female, and these are their kids. So we know that the purple means that the male is affected. So the male has a widow's peak, and the female doesn't. So the male is going to be heterozygous, and the female is going